In this video, we can talk about the layers of gut wall and its histology. Our gut wall is made up of four layers: mucosa, submucosa, muscularis, and serosa if it is an intraperitoneal organ, adventitia if it is a retroperitoneal organ. Let's see the image to get more clarification. This is the mucosa layer, which composed of epithelium, lamina propria, and muscularis mucosa. Then comes the submucosal layer. In this region, we having submucosal plexus, and in submucosa, we also having submucosal gland. These submucosal gland can be undergo secretion with help of submucosal plexus. This is muscularis propria, which contain a two layer. One is the circular layer over here, and another one is longitudinal layer over here. Between these, we have the aerobract plexus or meandric plexus. Then comes the adventitia or the serosa layer. If it is a retroperitoneal organ, it will cover last layer by adventitia or outer layer by adventitia. If it is a intraperitoneum, the outer layer covered by serosa. So let's see other image. In this image, we can see in submucosa, after this is mucosa, then comes the submucosa. This is the lumen over here, and after the submucosa, here we have the mucinous plexus or the submucosal plexus. Then comes the muscularis propria. Between the circular muscle and the longitudinal muscle, we have the aerobract plexus or the meandric plexus over here, and then is come the adventitia or the serosa, depend upon the where it is if it is intra or the retro. And we also have one more image. Like in this image, we can see this is the mucosa layer, and this is the part, and this is the submucosal layer, and here we can see a submucosal plexus, and here we have two muscles, inner circularis, outer longitudinal muscle. Between that we have the meandric plexus. In case of ulcer, it will be extending into the submucosa and inner and outer muscle layer. If it is erosion, it will affect only the mucosal layer. In GI system, the electrical rhythm is provided by the interstitial cell of Kajal, like we see in SA node. But there is a small difference. These have a slow wave. This means that if there is an electric potential graph, if we need a certain potential for the muscle to contraction in the GI, there will be a slow wave like this. If the stimulus is received on this point, it will be very weak, like this. But if the stimulus is get on this area, it will be high. So the muscle will contract easily on this area. We have 3 waves of slow wave in stomach, and 12 slow waves in the duodenum, and 8 to 9 waves in the ileum. In this image, we can see a blue one, which will be a meandric plexus, and this is the interstitial cell of Kajal associated with meandric plexus, provide the pacemaker to the enteric system. So let's talk about the histology of digestive system. Esophagus is made up of non-keratinized stratified squamous epithelium. Non-keratinized means they don't have keratin. So since there is no deposition of keratin, so it can easily permeable to fluids. If there is a keratin deposition, there cannot a fluid passage, so that portion will appear dry. Since we know that our oral cavity and esophagus is always wet, it is because it is non-keratinized, and it is stratified means they have more layers, and it is squamous. Upward one-third of the esophagus is striated muscle, means the skeletal muscle, and middle one-third it will be the mixed of skeletal and smooth muscle, and lower two-third will be the smooth muscle. Like the upper portion is skeleton, and the rest portion is two-third, one-third is the mixture of the skeleton and the smooth. Then comes the last one-third is the smooth muscle. In case of a stomach, we have gastric gland, which is represented here. Let's see another image to get more clarification. In this image, we can see a mucosa here. This is up to this portion. So, in this place, the surface epithelium will be the pit of the gland until up to here, as shown here. This is the pit of the gland, and this is the gastric gland. And this gastric gland contains many cells. One is the parietal cell, which secretes the H plus. Other is the chief cell, which will release the pepsinogen. Like we see here, the parietal cell goes and binds to the pepsinogen, which is secreted by the chief cell. It may converted into the pepsin, which is the active form, which will cleave the protein. In case of the duodenum, we have villi, and this villi also have the microvilli over here. So it have microvilli and villi. We have high absorptive surface. Duodenum contain a Berner gland. This gland is present in the submucosal layer, and it also crypt of Leberkin, which contain a stem cell for the replacement of enterocyte and goblet cells. It also having a parent cell which help to secrete the defensin and lysosome and tumor necrotic factor to fight against the microorganism. This is our innate immune system. In the jejunum, we have villi, and it also having crypt here, and the pilla circularis also present. This structure is also visible in the distal part of the duodenum. In the ileum, we can see a pear patches over here on this side. This is the lymphoid aggregate in the lamina propria of submucosal layer. It also having a pulca circulari, which means a fold in the mucosa, which is seen in the proximal part of the ileum. It also having a crypt of Leberkin and largest number of goblet cells in the small intestine. This is the place where the large number of globular cells are present. In colon, we can see a crypt. Here we can see a abundant number of globular cells with no villi.